video will include parts of Chapter 24, which is on quantitative genetics, and also part of Chapter 7, going back to linkage. So up until now, we've talked about examples, what we call Mendelian inheritance. That is when two different genotypes give distinctly different phenotypes. A lot of traits, however, and particularly human traits, are not Mendelian in nature. And so these are called continuous traits or quantitative traits. An example is human height. We all know that human height is genetic, and if your parents are tall, you also tend to be tall as well. However, this is not this distinct 3-to-1 ratio. This is a quantitative trait. And quantitative traits tend to give bell curves in the population. Now, the reason why we have a bell curve is that this phenotype is actually determined by many different genes. So the genes that contribute to traits like this are called quantitative trait loci, or also called polygenes. Another example in humans of continuous variation is actually skin color. So we all know that the amount of melanin that is made by an individual varies tremendously. And this is just a picture of President Obama. His mother had a lot less melanin. His father had a lot more melanin. And he is somewhere about halfway in between. So this is the normal continuous variation, and I just want to point out that the same trait can also have alleles that are discontinuous in nature. So for example, an albino individual has no melanin at all, and so this falls into two distinct phenotypes, having melanin and not having melanin. In contrast, there are 378 genes that are known to be involved in normal melanin production. And the number of distinct genotypes, if you only have two different alleles for each of those 378 genes, gets really huge. So I'd like to start off by talking about a very simple example of polygenes or quantitative traits. So this is looking at wheat seeds, and it's looking at how red the seed is. So for this trait, it turns out that there are two genes that contribute to how red that seed is. Each dominant allele contributes one dose of redness, and each recessive or mutant allele does not contribute a dose of redness. So you can have individuals with 4 doses of redness. It makes very dark red or purple wheat seeds, or you can have no doses of redness at all. Now, just as an example, I'm going to do this cross. And so if you cross a individual with all recessive alleles to one with all dominant alleles, you will get a dihybrid individual. And so this one is sort of intermediate in their seed redness because it's got only 2 doses out of the 4 possible. If you cross this F1 generation together, we've done this dihybrid cross many times before, and this is just 2 ways of doing it. Your textbook actually uses a branch diagram and looks at both of these genes independently. Another way to do this is just a standard 4x4 Punnett square. This notation is slightly different than what we're using, but it gives you the idea. And so the idea is that you can get different phenotypes in the F2 generation, and the number of doses of the pigment gene will vary. Very rarely will you get all 4 pigment doses, and very rarely also will you get no pigment doses. Most often you'll get something in between. And in fact, if you graph this, what you see is that the most common is actually this intermediate phenotype of only 2 pigment doses, and the most rare are these 2 extremes. And what I'd like to point out is that even though there's only 2 genes involved here, this starts to approximate a bell curve. And if you also have a little bit of environmental influence here, you really will start to get a bell curve with even 2 genes. Now, it turns out that the more genes that are involved in determining a trait, the better that phenotype will actually approximate a bell curve. And so if you only have 1 gene involved, you get these 3 genotypes. They tend to be distinct phenotypes. However, 2 genes involved, you start to approximate a bell curve. 5 genes involved, and it's looking more and more like a bell curve. So it turns out that a lot of the most difficult diseases facing humans these days are influenced by multiple genes. So these would be quantitative traits, things like diabetes, especially type 2 diabetes, cancer, mental illness. We know that all of these things have a genetic component to them, but they also have strong environmental components. And not all of the genetic loci that are involved in the susceptibility for these genes are actually equally important. So there may be, 2 or 3 genes that are very influential for diabetes, but then another 10 genes that have only minor effects, for example. So next I'd like to talk about how we actually locate genes that confer susceptibility to complex diseases such as diabetes. So in the linkage chapter, we talked about this traditional approach, which relies on looking at families. And so this really looks at, who in the family has this disorder and whether there may be any SNPs that are linked to it so that we may actually locate which gene is giving the susceptibility for this particular trait. So this works really well for simple Mendelian traits, and it does not work so well, however, when we look at much more complex diseases. So I'd like to tell you now about a different kind of study that is very useful for looking at the inheritance of quantitative loci for complex diseases. This study is called a Genome-wide Association Study, or a GWAS study. And what this does is it uses a DNA chip to look at thousands and thousands of SNPs in one individual. Now, we will talk later about the technology involved in a gene chip, but suffice it to say for now that we can look at thousands, even up to a couple of million SNPs all at one time. And rather than looking at the inheritance within a family to see whether that particular SNP, along with the disease, was inherited in your particular family, a GWAS study simply looks for a statistical association of a particular SNP with a particular phenotype. So, in other words, if you have a particular SNP, you are more likely to have a particular phenotype. It's looking for correlations here. So I'd like to give you a very simple example of how a genome-wide association study might be done. So if you wanted to study type 2 diabetes, you might get 2,000 people who have type 2 diabetes and compare them to 2,000 people that do not have type 2 diabetes. And then you might go in and measure a whole bunch of different SNPs in every single individual with the DNA chip. For every SNP, then, a statistical test would be done to determine whether that SNP was more likely to be found in individuals with diabetes than in the control group. So here's an example, just 10 individuals to give you a feeling for this, looking at 3 possible SNPs. And so each one of these individuals, it's indicated whether they have diabetes or not. And it turns out that 2 of these SNPs have no association with diabetes. So SNP1 and SNP3 have no association with type 2 diabetes, but SNP2 does in fact have an association. So if we look here at the genotypes of the individuals who have diabetes, what you'll notice is that these individuals with diabetes all have at least one A allele in that SNP2. In contrast, these individuals without type 2 diabetes, a bunch of them have a GG genotype. Now, importantly, in this kind of test, remember that there are other genes that are involved. So this particular SNP is not the full story. It could be that some of these individuals, for example, number 7, who does have an A allele but does not have diabetes, possibly they have a genotype at a couple of other loci, which gives them a decreased risk of getting type 2 diabetes, and so that's why they don't have it. In a GWAS study, usually what people do is they look at markers that are spaced throughout the entire genome. And so here you can see an example of a GWAS study for Crohn's disease. In the second panel here, you can see each of the chromosomes is listed. And so using SNPs that are arrayed throughout chromosome 1, it turns out that there's one location that reaches significance for being associated with Crohn's disease. There's another one that's very low here. This may be background, or it may be something that has a very small effect on the genetic susceptibility for Crohn's disease. So GWAS studies establish linkage between a particular SNP and a susceptibility for a disease. Now, you might assume that that SNP is actually the causative thing that makes someone susceptible for a disease. And it is true that sometimes the SNP is the actual cause of the susceptibility. If that is the case, that is actually called a functional SNP. Often you would find that that SNP occurs within a coding region of a gene, and that is the one that is actually making this trait change, giving the susceptibility for this disorder. However, this is not always the case. So sometimes that particular SNP is only linked to the actual DNA change that causes the trait susceptibility. It turns out that groups of SNPs tend to be inherited together. And so if you have a group of SNPs that is inherited together, it is called a haplotype. When a SNP is within a group of SNPs making up a haplotype of SNPs, it turns out that you can measure any of those SNPs within the group, and any of those SNPs will tell you whether you have the functional SNP or the one that is actually conferring the trait change. Now, this is a little confusing, so I'd like to go into a little more detail about it. So, humans are all related, and because of the general relatedness of humans, we have these groups of SNPs that tend to be inherited together. So if you have an ancient individual who actually had this causative variant that gives susceptibility for a disease, so they contain a functional SNP, and throughout many generations, you get lots of interbreeding and you get lots of crossover events so that this particular ancient DNA sequence is now broken up. Nevertheless, you have these regions of the DNA that tend to stay together, and these are called haplotype blocks. Now, within this haplotype block, this is a group of SNPs that will all be staying together. And so here you have a mutation in a particular gene. This is the mutation that may give this individual susceptibility to type 2 diabetes, for example. However, there are all these other SNPs that are linked to it, and these SNPs are usually found together with this particular causative or functional SNP. So you can measure any of these SNPs and predict whether that functional SNP is present. So the reason you actually get haplotypes is because chromosomal recombination or crossing over is not actually random, as the early chromosome mappers thought. So what you actually get in reality is there are a number of places along the chromosome that tend to get recombination. So here's a spot here, here's another spot, right? Here's another spot. And you tend to not get recombination between these recombination hotspots. So here's another example to look at this idea. Here you have regions along a chromosome, and the locations of a bunch of SNPs are indicated. And so here's SNP 1, 2, 3, 4, 5. These all fall within a haplotype block. So what that means is recombination tends to not occur between these individual SNPs. Here's another haplotype block. These SNPs 6 through 10 tend to stay together, and so you do not get recombination between these individuals. Now, there is some kind of disease we are following here. SNP 8 is actually the functional SNP. That is the change in the sequence that causes the susceptibility for some disorder. And let's just assume that the G genotype gives the susceptibility for the disorder. You could potentially measure the SNP on SNP 8 and figure out the susceptibility for this disorder, or you could measure the SNP on SNP 6, which is in the same haplotype block as SNP 8, and also know whether that individual has susceptibility for the disorder. So if you measured SNP 6, you would actually look for an A, and that would tell you that this individual most likely has a G at SNP8 position and therefore is susceptible for the disorder.